Hi everyone, I'm here with Christie Wang, and
Hi.
This is the Alex Jafari Research Show. Christie is a Siemens regional finalist, and she went to Pittsburgh. She's also a high school award recipient for the American Academy of Neurology, and she went to New Orleans, which is major. And she also got, I believe, 3rd place at LICEP, correct?
Yes.
Great. Christie, tell me your project title and what did you do?
Well, my project was entitled Cold Shock Protein, a new target for attenuating ischemic stroke. Basically, I discovered that a protein, a cold shock protein called cold inducible RNA binding protein, or CIRP, caused inflammation in the brain. And basically, targeting it can be a new effective way to treat stroke patients or people who stroke.
So that was a purpose, essentially,
yes,
right?
Okay,
cool. So, I know that you did your project over the summer, mainly for how many years did you work on it?
I worked for 2 summers at the Feinstein Institute with my mentor, Dr. Yang.
Okay.
So yeah, my project is a cumulative project.
Cool. So can you give me a little day-to-day type of experience, over there?
Yeah, well, I worked 6 days a week, Mondays through Saturdays, from about 9 a.m. to 6 or 7 p.m., depending on the day. And basically, the mornings were usually experiments, and the afternoons were usually working on papers or, doing statistical programs and whatever, running programs. And also, yeah, so each day was basically just a new experiment or a continuation of the previous day's experiment, depending on the project. Any
particular skills that you would say research students who are interested in natural science would need to do a project like yours?
I honestly don't think you need skills per se. I think you can develop those at your time at the lab, but I do think you need an interest in biology, an interest in your project to stay dedicated, to enjoy it, I guess, so you will stay motivated and work hard throughout the whole entire summers to get the most out of your summer. So usually if you're interested in biology, you do well in biology. So I guess strong science skills and motivation is what I think.
So we're here again, and I'm just going to ask you a few questions about the general procedures you employed. So what was the general methods in the most layman terms possible?
Well, I basically measured protein levels or enzyme levels and how they're affected by other conditions such as hypoxia or adding a recombinant CIRP. And the actual methods I used were real-time PCR, Western blot, ELISA, cat-specificity assays. I also transfected cells. So that's basically it, just a lot of assays running them over and over.
Cool. So here on the board we have this very, I think, elaborate but also succinct type of diagram that you made. Is it your results, basically?
Yeah, this is basically my results, the pathway I discovered, basically how you can target CRP, where you can target it, that kind of thing.
So can you just give a little explanation?
Oh yeah, sure. This big gray box is the BB2 cell, which is a microglia cell in the brain. Those cells usually are linked to a lot of inflammatory processes, so that's why I use the BB2 cell. And then when it's placed in hypoxia, CIRP is released from the nucleus. It's a nuclear protein, so it's normally within the nucleus. It's released from the nucleus. Ooh, fancy. Okay. And then CRP is released outside of the cell. So normally in normal conditions when CRP remains within the nucleus, it's in almost all of your tissues within the body. But once it's released from the cell, it becomes harmful. So here, through autocrine signaling, it's accepted through a CD14 receptor, which according to literature has been known to accept a variety of other inflammatory ligands. So I decided, I tested it and obviously it worked. And then after that, this triggers the translocation of NF-kappa B from the cytoplasm to the nucleus, which is another hallmark of inflammation. And then the presence of NF-kappa B within the nucleus causes TNF-alpha to be released from the nucleus and from the cell, causing inflammation and injury throughout the brain, which is basically your stroke.
Cool. So this was your general results, the conclusions that you made.
This is basically my whole project.
Great. Okay. Tell me your overall thoughts about the whole research experience over the past 4 years.
I think it's really worthwhile. I think it's something that's important to stick to. It teaches you hard work, dedication, organization skills, even study skills. I think it's really helpful throughout high school, and I think a lot of people who have graduated have also found that it's extremely helpful going into college and also even into your work experience.
All right. Okay, cool. And so the most memorable research experience that you've had?
I think it's probably the competitions that I've been to, whether they're at LISAF, which is in Syosset usually, or even Carnegie Mellon, which was at Siemens, or New Orleans. Just all the little trips, meeting people, and I think like getting to know people is something really memorable about research.
Cool. So what would you have like as advice for upcoming research students in the program?
I'd say just stick with it. Don't quit. I don't want people to think about quitting, especially sophomore, junior year. But I think if you stick with it, you'll find that it's worth your time and it's worth your while.